One of the pleasures of my job is, as Director of the School of Public Health, I should say I'm Neil Ferguson and I head the School of Public Health where Victoria works, but one of the pleasures of my job is introducing evenings like this. They mark the promotion of someone to full professor in the university, a very significant step in their academic career, and inaugural lectures are a way we celebrate that achievement, but also showcase the unique skills, contribution and interests of new professors in the university. Victoria is a Professor of Medical Statistics and Trial Methodology. She's also my close colleague as head of the Imperial College Clinical Trials Unit, a role she took over about the same time as I took over the School of Public Health, I reminded myself earlier. And it's been a real pleasure to work with her. She's going to take us on a little journey through her professional life with a little bit of her personal life on one side and motivations, and talk to us about the contribution of new advances in statistics to more efficient trial design and the better, therefore, testing of medical innovations, treatments, and the like. So it's a real pleasure to introduce you, Victoria. You said you might overrun by a couple of minutes, so I'll keep my remarks short and sweet. We'll have questions at the end from the audience and then finish off with Deborah Ashby, previous director of ICTU and the School of Public Health, giving the vote of thanks. Go ahead.
Thank you very much, Neil. Oh gosh, this is very loud. I think. Right. Okay. Yes. No. Thank you, Neil, and thank you all so much. I don't know why it's forwarding on. Is that possible to not automatically forwards? I'm sorry about this. Some technical hitches. You've seen what's coming. That's very odd. We had a bit of a runaround. So we're okay from here. Hopefully that's it. Okay. So I'll just first of all tell you about. I wanted to welcome everybody online and also in person. It's really lovely that you've taken the time out to be here with me this evening. I also wanted to thank Imperial College for promoting me to a chair in medical statistics and trial methodology. And also to say that Imperial's been my academic home now for 8 years, and it really has been a supportive environment. You'll see that all of my research is collaborative. So as well as supporting me, it's given me a wonderful opportunity to connect with literally world-leading clinical academics as well as world-leading trial methodologists. And that's been able to shape the work that I've been able to do. I'm going to be talking about smarter trials. And so that's trials where we want to do things a bit better because actually we're asking more complicated things from trials. And smarter trials, there's a kind of tendency to want to do them faster and cheaper and quicker. But actually it's not just about that. Smarter trials are about doing better science. So actually we get the right answer from these trials, and we can do that, answer more relevant questions. And so I'm going to be showing you a little bit about how trial methodology can help us with that. And I was aware that not everybody is a trialist here. So I was going to do a quick explainer of which has started here. Okay. So a randomized control trial is the most robust way we can work out whether or not an intervention is working. And that might be anything from a drug or a vaccine, sometimes even just the way that we're looking after patients. And at its core, it's beautifully simple. We've got our promising interventions that we want to trial. We've got those patients needing those interventions. And if we can randomly allocate to groups, and we can conceal, oh, that's going the wrong way now, and we can conceal that randomization, what we do is we remove selection bias. And that means that we end up with highly comparable groups, such that we can follow those groups over time. And then any difference in those health outcomes between those groups, we can robustly attribute that to the intervention that we're studying. Now, the thing about trials is actually it's not just selection bias we need to be worried about. There's lots of other ways that bias can creep in, all the way from the very sort of this design until we report that trial. And so it's a key part of us role, as trialists to minimise the introduction of that bias. Because if bias gets in, what we're basically saying is we're going to get the wrong answer. Now, this is what we call a parallel two-arm trial. It's an absolute classic. It's been with us for about 75 years. And we've just done a recent review of trials in neonatal units, and actually over 90% of trials are still using this design. Right? So that's not that there's plenty of other trial designs out there, but like I say, this one is very popular. But the way we're doing trials currently has got huge challenges. They're taking a lot of time and a lot of money to do. So our recent data shows that the average UK academic trial costs 1.4 million. They're taking on average 4.2 years to conduct, and actually hardly any of them are recruiting as planned. So trials are under a lot of pressure. We're asking increasingly complex questions with them. And in order that we want to do that robustly, we need trials to be more efficient, more reliable. We want them to cost less. But most importantly, we want them to be less burdensome to deliver. And this is where trial methodology can help us, okay? So trial methodology is basically, it's a science into how best we design, conduct, analyse, and report clinical trials. So over the last 20, 30 years, actually, we've made some really wonderful advances, certainly in adaptive trial designs, and also for the promotion of Bayesian analysis in trials. But as a trials community, we've been pretty slow to take these up. And that is, I would say, until now. Things are really beginning to shift for us. So I'm going to introduce you now to a little bit to what is changing. Traditionally, the way we approach a trial was we make a number of assumptions at the start, and we design that trial. We then conduct it over our sort of average of 4.2 years. So by the time we get to analyse and conclude from that study, we're kind of making a gamble. We're gambling on the fact that all of those assumptions we've made sort of 4 to 5 years previously are still holding. And if they're not, we might not be able to answer our original question. So adaptive trial designs offer us a different approach. They sensibly look at the data as we go, and then we can change the trial. And if we're changing things based on the data, we can make it much more, we're much more likely to answer a reliably answer a relevant question. It comes with a strong caveat, however, that we need to pre-specify what we want to change. So some of you might be wondering, what is it we want to change? And others of you may have heard of many fancy adaptive trial design names, like MAMs and platforms and baskets and umbrellas and things. And actually, I don't think those names are terribly useful. I think with adaptive trial design, we're much better to be able to talk about what is it, what are the adaptive components, and put those components together. So the kind of things that we might want to do is we might want to drop a treatment or stop a trial early, because we might, we know that the treatment's working, or maybe that it's never going to work. We might want to change the dose of that treatment or add in new treatments to that trial. The other thing we might want to do, we might want to increase our sample size so we don't end up with an underpowered trial. Or we might want to change the population, because we might realise actually it's only working in a subgroup of these patients. And the other thing we can do is we can change the way we allocate patients to arms as well. So all of these adaptations, what they're trying to do is they're trying to target the right intervention, the right population, and also stop when we have enough evidence. Now I'm hoping we can move on. If that's possible, to move to the next slide. Okay, marvellous, right. So what we do, when we want to choose maybe one or more of these adaptive components, and then we can take those, and that's where the statistical design is, which we often do through simulation. So we work out at what points we should make these decisions and what points we should change. And we do that through optimizing our trial operating characteristics. and then we end up with different trial designs. But the key thing here is that we're designing those adaptations in. I'm so sorry, is it working? Right. Because, right, we can't just make stuff up as we go. Because if we make stuff up as we go, we end up introducing bias into that study design, and then literally it's not fit for purpose. So we can see methodology matters. We can use it to make trials more efficient, to get more reliable results from those trials, and ultimately what that's doing is that's offering patients better outcomes. So I'm going to tell you now how I ended up in trial methodology, and also how I ended up at Imperial. So I'm the youngest of 4, and I grew up in a fairly boisterous family. My dad was a physicist and a researcher for the government, and he was a lifelong civil servant. And I love this photo of him because he looks like a lifelong civil servant. And stood next to him is my mum, and she was a stay-at-home mum that looked after, honestly, us 4 brilliantly. And I was thinking about my education and what an unremarkable start I had to it. So I was actually quite a sickly child, and when I got to 12, I was actually off school for a whole year. And I really wasn't on any trajectory whatsoever to getting any GCSEs. And then when I went to go back to school at 13, my parents took a surprising decision, and they sent me to this beautiful school on top of a hill in the South Downs, and it was wonderful. I was suddenly in this wonderful place of learning. I had the most enthusiastic and dedicated teachers, and I had to concentrate in every single lesson. So it was here that I really discovered my love of science and maths, and I ended up taking my maths AO early, and I got my full set of GCSEs from it. So a similar time, I was just enjoying maths, and I had another love in my life. And that love was for theatre. So I spent all my time, I didn't read books, I read plays, and I threw myself into my local amateur dramatic group. And I also joined the Chichester Festival Theatre. And there were 2 particular highlights for me. One was being in the amateur premiere of A Pack of Lies, which is a true life MI5 story. And the other one was getting the opportunity to perform at the National Theatre in London, because we had got through to the finals of a youth theatre challenge, so that was great. So when I finished my A-levels, I decided that what I wanted to do is I wanted to study technical theatre and design and stage management at Mount View Theatre School. So I loved this. I got the chance to design sets, which I really enjoyed. I also got the chance to learn how to build them and paint them, and also to learn the craft of stage management. Okay, so that was like working backstage, making sure everything's working well out the back so that the show can go on. So we practiced our skill on a couple of many actors, very talented actors, and these are a couple you might have heard of, so with Amanda Holden and Eddie Marsan. So after Mount View, I worked in professional theatre as a stage manager. So I got to tour around the UK on various different productions, working on things like Anton Chekhov at Chichester Festival Theatre, to the Chippendales in the Strand Theatre. So this career might look quite far removed from what I'm doing now. But actually, there is an analogy here with trial methodology. Bear with me. So the analogy here is actually trial methodology is the stuff behind the scenes that you don't see, such that the trial works. And it's similar with the stage management. You know, that's the stage management's all the stuff behind the scenes, such that things can work well out front. Okay, so I was enjoying myself here, but I was missing an academic challenge. So I decided to go back to university to study for a degree, and I chose to go to Cardiff, and I chose mathematics just because that was the most the thing that I was most comfortable with. And I had the full intention at that time of going back to theatre after I'd done my degree. But actually, I did enjoy studying. And then at the end of my degree, I saw an opportunity to do a PhD in medical statistics. And that really captured my interest. It was for the first time, to use this large linked data set for all the accident emergency departments around Wales. It's going to be, and I was going to get the opportunity to analyse this data set. Now, big data. So unfortunately, that large linked data set never turned up for the whole of my PhD. And actually, it was a really concerning time because I was hanging on waiting for it. But and there were no checks on PhDs in those days. You didn't check you were on, you're doing okay. You just hoped you had enough by the time you got to the end. So, but I was very fortunate. I got to do some spatial epidemiology. I was exploring environmental exposure and breast cancer, and that was enough to award me my PhD. And by now, I was fully committed to medical statistics because I just loved the sort of maths with the application of healthcare. So I was very lucky. My first role after my PhD was at the Oxford for the Centre for Statistics and Medicine, working for cancer research and being led in that research by Sharon Love, that I'm hoping is here tonight. Yes, she is. Well, I can't quite see. Anyway, hello, Sharon. And so the group was led by Doug Altman. Many of you who do know Doug know he was absolutely dedicated to improving medical research practice. And he really has shaped a lot of the way that we do good practice today. I also met Leslie Smith at the time, and we worked on for several years on different systematic reviews. And what was good about that was actually that gave me a really good foundation in trial design and analysis. But one of the main things I took away from the CSM was around doing methodology well, because it wasn't obvious to me when I went in, but actually not all research is good. And Doug's work highlighted this and really highlighted things like if there's weaknesses in the methods, it's really fundamental. If we don't get the methods right, we get the wrong answer, and then we harm patients because that's what we're using to make our decisions for those patients on. So a little segue here, because after I joined the CSM, I then had two lovely children, and I moved back to Hampshire to be closer to my parents and my sister. And hopefully my dad has been able to join online tonight. And my Auntie Lizzie is here as well, as are my two lovely children, Florence and Leo. So when I had children, that was the start of 14 years of part-time work for me. And I actually only returned to full-time work when I joined Imperial. And when I did go back to work, first of all, I went to the Drug Safety Research Unit in Southampton. And my work here was really focused on post-marketing surveillance of new drugs. So that's when we've trialed a drug, it's got a license. When it goes into the general population, we want to signal any adverse drug reactions that we haven't seen in those clinical trials. So there was a lot of focus here about statistical methods on which, on how to signal those adverse drug reactions. And in the early stage, I met Odile Sozu, and she encouraged me to write my first statistical methods paper. And we called it the WASP test. And it was, what was special about our test, and we're working with Professor Stephen Evans as well at the time, we didn't need a large data set. It's a very efficient method. We could just do it on an exposed cohort only. And there's so much to say about this, but I'm not going to say any more, other than we followed it up with papers over the years, first of all showing how to implement those methods, and then also refining those methods a bit. And I have to say, it takes years actually for methods to get into practice, and I'm delighted to see that actually our WASP test is being used. So I was happy at the Drug Safety Research Unit for a number of years, and then I saw a new senior statistician post going for 2 new biomedical research units that were being set up at Southampton. And this was a really exciting position for me. It was back in a university, embedded in a public health group, and then working on a whole range of studies, all the way from laboratory studies through to clinical trials. This gave me the opportunity to have my first senior roles on trials. And I first working with Professor Graham Roberts and Professor Hassan Arshad on their allergy prevention trial in babies. and also working with Professor Saul Force and Dr. Katrina Cathy on their trial for looking at treatment during an exacerbation for cystic fibrosis patients. Now, Graham and Saul were both involved in these new biomedical research units, in the management of them, and Oxford University were having an open network day to sort of share their learning and recommendations for developing clinical trial databases. So we went along thinking this will be a great, there might be something here we can bring back to Southampton. And it was such a useful day. And we were reflecting on this on the train on the way home. And that's when Saul said to me, Victoria, you could do a statistical network, something like this for statistics. And so that was the sort of catalyst for the NIHR statistics group. And Saul put me in contact with NOCRI, and NOCRI said, yes, we're very happy for you to develop a group like this. And so we set up the NIHR statistics group. First of all, it had wanted to have the aim to share good practice and learn from one another. But also it had the important aim for me, which was raising the profile of statistics within the NIHR, because suddenly we have a name. And it was very clear early on that I think within 2 years we had 300 members, so this was going wider than NIHR statisticians. And we had 4 very active working groups. And then we started developing best practice papers together as well. So I was involved in 3 of those. First of all, one for looking at improving the design analysis of laboratory-based research. And then we had sort of sister papers, one about embracing model-based designs for early phase clinical trials, and then sort of demonstrating the implementation of those. So just want to say that the group is still thriving massively. Actually, Professor Dawn Tier and Katie Bunce took it to the next level, and now it's under new leadership of Liz Henson and Andy Vale. So my next move from Southampton was to King's College London, and that was where I had my first academic role as a senior lecturer. And I was working with much more focus in clinical trials. I was working on feasibility all the way through to phase 3, and also learning a lot from Caroline Murphy, who was the operational director at King's. And I was an affiliated King's Clinical Trial Unit member. So one of the trials I was working on was Professor Catherine Smith. And Catherine wanted to study whether or not anakinra would be a helpful treatment for pustular psoriasis. Now, pustular psoriasis is a rare condition. It's horrible because you get these pustules on the hands and the soles of the feet. And actually, there's very limited treatment options. What treatments do work, unfortunately, have nasty side effects. So there was a real reason, it was a real area of need. Now, Catherine believed that there was, mechanistic reasons that anakinra would work in this population. But it did mean that we didn't have any proof of concept data. We didn't have any safety data. And we weren't sure how to measure the outcome. And in trials, we need to show that it's actually getting better. So there was no validated outcome. So the trial methodology question here is, of course we could take many years to do this. But could we do this? How efficiently could we do this? So if we look at the data as we go, then we can test some of these things out. And so this was my first adaptive trial design. We started out with 2 primary candidate outcomes. One was a validated score, but it was validated for psoriasis. And the other one was the idea that we could count the pustules on the hands and the soles of the feet. So we started out with those. And when we had set up an independent data monitoring committee, they had very close reviews of the first patients into the trial. And then at the end of stage 1, we asked the IDMC to make 2 decisions for us. First of all, could they make sure that there was a proof of concept, so is there a signal of efficacy? If not, we should stop. If there was, could they choose our primary outcome for us? Now, when you're choosing your primary outcome halfway through the trial, you can't do that on a treatment effect because otherwise you'll introduce bias. So we had given them guidance around looking at the distributional properties of those outcomes. And as it turns out, you can't reliably count pustules on hands and feet. So we went forwards with the validated psoriasis score. Now, we finished this trial, and unfortunately, anakinra wasn't demonstrated to be an effective treatment. But sometimes it's as good to know what's not working as what's working. So I was doing a lot of trial. I've talked a lot about showing about treatments working, but actually a really important part in trials is also to make sure that we're not doing more harm than good. So we have this risk-benefit that we need to balance. And this was a start, and I was really struck by my drug safety research unit days. We'd gone to so much effort to look at, how we were analysing the data. And here in trials, we were literally just tabulating it and then sort of subjectively assessing it. So that was began a new research stream for me to be interested in drug safety and improving the analysis of adverse events. And then I was joined by a PhD student who had huge commitment to this, Rachel Phillips, and Rachel is now a senior colleague with me at Imperial. And with her phenomenal drive and energy, we've been working on this for nearly a decade together. First of all, around articulating the problems, then sort of looking for some solutions, and then more recently for getting some international collaboration on this. And Rachel's now leading an international forum for this. So I was enjoying myself at King's for a while, and then I saw an opportunity, and it was to work with Professor Deborah Ashby at Imperial Clinical Trials Unit, and also to have a leadership role of the stats team. So IC2 was co-directed at the time by Deborah and Professor Neil Potter, and I saw this as my dream job, to be fully embedded in a trials unit and also to be working with Deborah. And I just want to say a huge thank you to Deborah. You've made me feel like a proper academic from the moment I stepped through the doors at Imperial, and to Neil, who has always constantly endorsed me and encouraged me in every role that I've had since I've been here. And one of the things I was first able to do was to recruit my first senior statistician and to help me on my trials. And I was so lucky, Dr. Susie Crowe. She's now, again, a senior colleague with me at Imperial. And she co-leads the stats group with Rachel. And what Susie brought me and my trials was this sort of methodological rigor about how she approaches everything. And especially with regard to missing data, because again, missing data is something where if we don't get it right, again, we can get the wrong answer, because if we don't handle that correctly. She's also been an early adopter of the Esteban framework, and she's internationally leading now in both. And again, what she's brought to the unit is she's made sure that as a CTU, we are also leading and adopting those best practices. So a couple of years ago, as Neil mentioned, I was promoted to a director of Imperial Clinical Trials Unit. And I just want to say a huge thank you to all of IC2 and the senior management team for being so incredibly supportive. I just wanted to say a special thank you to the operational director, Daphne Babelis, literally who I learned something from nearly every day. So at a similar time as becoming IC2 director, I also was working with Professor Greta Raitt from UCL and Professor Richard Hooper from Queen Mary. We put together a successful NIHR infrastructure grant for a new research support service. So that's a national service, and it's sort of delivered across 4 universities. So we were successful. I co-direct that with Greta, and I wanted to say a huge thank you to the operational director, Gareth O'Brien, who literally makes mine and Greta's job so very easy. So I'm just going back to trial methodology. I want to give another example of a challenging design question and how, again, trial methods can help us. So when I first joined Imperial, I was working with Professor Sejal Sirglani, and she wanted to study a really important subgroup of children, and these are who've got asthma. So these are children that, despite optimal therapy, they still keep having repeated asthma attacks. And so that makes them very high risk of poor outcomes. And there is an effective treatment that's called omalizumab, but it only works in about 40% of children. And Sage, again, for mechanistic reasons, believed that mepolizumab may be help these other children. Now, we couldn't trial mepolizumab against placebo, because that wouldn't be ethical. So we needed to trial it against omalizumab. And we don't always want to show things are superior. Sometimes we want to show that they're not inferior to. Now, non-inferiority trials generally have larger sample sizes. And here you can see we're in this small sample size setting where, it's going to be challenging. And the other slight challenge we had with this was because we wanted to show that asthma attacks were going to be reduced over a 12-month period, we couldn't use any kind of adaptive trial. And so I just want to say, non-inferiority trials are commonly poorly done. All right? The critical component when we design a non-inferiority trial is the width that we set the margin. Because if we set it too wide, that's the thing we're testing non-inferiority against. If we set it too wide, then we can end up drawing harmful conclusions. And then again, we harm patients because we say these treatments are similar and then they're not. So I was very aware of that with the treat and being only able to recruit a sort of a maximum 150. And I didn't want to do the common thing, which everyone does, which is just make those widths a bit, the margin wider. So I realized we were never going to do this in the frequentist setting. So the solution here was to use a Bayesian framework. And we started from the premise of, actually, the sample size is what it is. We can't do anything about that. We're not going to be able to recruit more than 100 to 150. But the Bayesian framework could help us in two ways. First of all, we could incorporate existing information, which we did through a clinician elicited prior. And that helps us with our estimation. But secondly, and much more importantly, is that we could change the inference framework. And we could talk about the results in terms of probabilities of non-inferiority against a sort of credible non-inferiority margin, so that we weren't in any danger of making harmful conclusions from this trial. So what I was navigating here is common in a common problem for non-inferiority trials. It's also made worse when we've got this restricted sample size. So working with Susie Crowe, Jack Elks, and some wonderful statisticians at the UCL, we took what I did for treat and we turned it into a framework. And this is literally hot off the press. I've just done the proofs this week. And the idea is that this will support better design for future non-inferiority trials. Okay, so I was at Imperial when the pandemic happened. That's obviously impacted us all in very many different ways. In trials, it impacted us. First off, everything kind of came to a grinding halt. And the trials we were doing, some of them went on pause, but some of them never recovered. So while it brought this disruption, actually, what the pandemic brought us was incredible collaboration and innovation from many clinical trialists. And now it has changed the landscape for us in trials, because we now know much more what we can do, and there's much more willingness to push boundaries and more openness to doing things a bit differently. So one of the successes for the pandemic was the wider adoption of platform trials. And there were a few before the pandemic, but they were very few. So platform trials are trials where we can not only answer multiple questions and start off with multiple treatments, but then, as well as dropping treatments when we find out whether or not they're working, we can also add in treatments as we go. And in the pandemic, platform trials were set up in remarkable time speeds and helped find effective treatments for patients. And they saved millions of lives, including the one that was led by our Professor Tony Gordon from Imperial for REMAP-CAP. So as well as rapidly finding effective treatments for patients, vaccines were developed and trialled in mind-blowing speeds. We had effective vaccines within 9 months of the pandemic starting. And I'm going to tell you briefly, very briefly, I promise, about a vaccine trial that was I worked on with Sourforce. It was led by Sourforce, called CovBoost. And there's a lot to say about this trial, but I'm just going to pick out one aspect of it, which builds on some of the work that Rachel and I have done about the importance of communicating harm. So I don't know if you remember, but by the start of 2021, most of us had either had 2 doses of AstraZeneca or 2 doses of Pfizer. And there were many other effective vaccines emerging onto the market as well. And the CovBoost trial was to determine which would be the best vaccine to give the UK population as their 3rd booster in autumn that year. Now, we were looking at 7 different vaccines. Actually, we had 9 different arms because some of them were at half dose. And it needed to be delivered incredibly rapidly. So it ended up quite a complex design with 3 different cohorts and control arms within those cohorts. And in short, what we found was nearly all of the vaccines worked. So when nearly all your vaccines are working, the question then becomes, well, are they any different with regard to how their side effect profile? And so we had 17 different pre-specified side effects that we were looking at. And I'd said the design was complex, so we ended up looking at pages and pages of these plots. And actually, these were stacked bar charts, so what everyone else was using. And we were trying to look across like vaccines, cohorts, time periods, and events, picking out the patterns. And I remember sitting with Saul, obviously online, because it was remote, and thinking, gosh, if we're struggling to work out what this is, what's the JCVI going to be doing? And the regulators, they've got less time than us to look at this data. So how are they going to weigh this all up? So what we needed something different here. We needed a simple summary measure that could sort of represent the total side effect burden. So hopefully I'm going to show you the power of a good visualization, which can aid interpretation. Okay, so we developed a plot, which we called a fingerprint for the burden of side effects. And what this allowed us to do, it allowed us to compare the vaccines directly. And it's simple because we've got all the harms on the outside, on the dial axis, and then each spoke is the proportion of the population that has had that event. And so the bigger the area, the more burden it is for these vaccines. And so in this particular plot, you can see Moderna and CureVac had a much higher burden of side effects than, say, half-dose Pfizer and the control. But I just wanted to demonstrate, because, using simple visualizations can really help us be able to communicate and also can help us with inference. And it's just something we do not do enough of in trials. We just don't take the time to think carefully about them. So we gave the JCVI this and a few other plots and all of the other data, and that's what they used to recommend the vaccines that you had in autumn 2021. So since the pandemic, I've worked on loads of stuff that I really wanted to tell you about and didn't have time, and really many interesting methodological challenges as well, including our first platform trial. But I just wanted to finish with highlighting 2 ambitious projects. These are about data-enabled trials. And so a data-enabled trial is basically a trial where we're trying to use existing systems in order to allow less burdensome delivery for staff, healthcare workers. So Collaborate is an adaptive trial that I'm working on with Professor Nina Modi. And in this, we want to answer 2 questions around feeding in premature babies. And the aim of the trial is going to be recruiting around 3,500 babies, premature babies. And then rather than doing that in a separate database, we're going to be using their electronic health records. And that's to save a lot of burden for all the staff. I'm also working with Dr. Tim Rawson as part of a new grand challenge for antimicrobial resistance. And the ultimate aim of this trial is we want to determine the optimal prescribing sequence for antibiotics for patients who are in hospital with sepsis. It's a very ambitious project because what we're aiming to do is we're aiming to fully embed this trial within the electronic health record system. So that is from the identification, including consent, to be able to risk stratify them, to be able to randomize them, and then to extract their data as well. So it is quite technical. We're also going to be comparing it to a traditional trial approach, so we can quantify the benefits of embedding versus our traditional trial approach. Okay, so I said that we want to do smarter trials, and we need them to be better, and we need them to be more reliable, more efficient, and cost less. And hopefully you can see from trial, if we use trial methodology, that can really help us with all of these things. And since the pandemic, we've done really well to sort of change the way that we do. We are much more open to embracing new designs and new things, new ways of doing things. But the really important thing is it matters how it's done. And hopefully, we've got to pay attention to the trial methodology, because if we don't get it right, we get the wrong answer, we harm patients. But I see our next big challenge is to make trial. Oh, sorry, one more thing. We're setting up a new NIHR Research Methods Academy, and that's got the sole aim of putting better methods into practice. So that we've just started this year, and hopefully you'll hear and see more of us. But regarding the final challenge that we need to do is to make trials less burdensome. Now, often we talk about data-enabled trials, about being cheaper, quicker, faster. And actually, I was thinking about this. I don't think it's not any quicker to do these, trust me. But what it is less burdensome for healthcare staff, and certainly for patients as well. We need to be more patient-centered. And that's where decentralised trials, which again, I haven't had time to talk about, but decentralised trials can help us. And I feel that we're just at the start of that journey, because it seems so obvious. It seems like the next step. But in reality, the national infrastructure is just not there to do this routinely or at scale, certainly when we want to do adaptive trials or anything that's got patient-centered outcomes. But I have to say, that's the things we're trying to do with Collaborate and Path. And then if we can do that, we really will have transformed trials. So thank you very much.
Thank you for a wonderful talk, Deborah, and I had no idea that you once were nearly on the stage. Now it's time for questions. If people can raise their hands, and we have two student ambassadors who will bring a microphone, and the first one off the mark was definitely there.
Victoria, thank you for sharing your life story and all your wonderful ideas of the design, delivery of your research experiences and clinical trials as well. I just want to ask, what do you prioritize when working with clinicians?
What do I prioritize?
Yeah.
That's such a good question.
It's an honor to ask the first question. Thank you for taking that question.
Yeah. Do you know what? Teamwork and collaboration is absolutely key. So it is fine, yeah, it's absolutely that collaboration. And hopefully that we're moving past the days where we're brought in at the last moment and treated a bit like a service, yeah. So it's that feeling for collaboration, that they want to truly collaborate.
That's a very diplomatic answer. Next one there.
Thank you very much. Quick reflection. I mean, you mentioned cost and speed and everything, and in a sense, the industry, healthcare and all the healthcare providers and industry have known about the cost and the complexity of randomised controlled trials. For 20, 25 years we've been talking about it. And then we all got excited, or I did, when we demonstrated during COVID that we could actually accelerate hugely. But my perception, since is that we haven't necessarily capitalised or built upon what we were able to achieve in those years. And so I wonder whether you could comment on that, and also why it is that maybe healthcare hasn't necessarily learned from other industries that have discovered agile methods, and I think particularly of, say, software engineering, that sort of started agile software development way back in around 2000, and as a to get over the sort of traditional waterfall approach that takes forever.
That's quite a lot to to thinking about the first question. It's really, I think I spent quite a long time with other colleagues talking about and thinking about why haven't we been able to do exactly what we did in the pandemic. But I think it was incredibly special circumstance. And I know that, as I said, actually all of our other trial work went off. So we were all working on one thing. I think that's the difference. At the moment, we're pushing many different things forwards at the same time, and actually just all focused in one area, we made a lot of progress. And also that it wasn't just the trialists, obviously it was the regulators, everybody was turning stuff around within hours, days, because we could, whereas now we're back to 6, 12 weeks, all those kind of waits. So I think I think it's not possible to recreate everything, but I would push back a little bit, because I think we have changed. Honestly, I feel that's what I feel, is that there's a change in that landscape of trials now because of people being so much more willing to adopt and progress things like the platform trials, innovative trial designs, and also the fact that we are wanting to do decentralised trials and data-enabled trials. So I think we are, it's just that we can't do them quite as well as we did then. Yeah.
Other questions? We'll get to both of you.
The UK would seem to be a world leader in formulating volunteer cohorts for data collection, like Biobank 15 years ago, and more recently, Our Future Health through the NHS. Are those tools that are of any use whatsoever in formulating medical trials?
I don't quite understand the question. Sorry, is that for the cohorts to be helping us in trials?
Well, yes. I mean, so we have available to us now data collection, medical data collection from millions. I mean, Biobank 15 years ago was 500,000, and Our Future Health is moving towards 5 million.
Yeah, so a lot of that, so that is sort of retrospective data. With the challenge with the trials is it's all prospective. So, and that's the challenge we have as well for scaling up. For less, for the data-enabled stuff, is doing that prospective recruitment, consent, and data collection. It's not, so the retrospective doesn't help us so much in that way. And even if we can access things, we're finding prospectively, there's quite long timelines, so it's not always very good, and you can't do much with an adaptive trial design like that. So.
Thank you. Mine coincidentally was related. There's a replication crisis for many medical trials, and we have built our current practice on a lot of flawed evidence. Your data-enabled trials, are you aware there is a sort of division in clinical practice? Are you able to use those retrospectively to re-examine some of the conclusions reached in previous trials? No?
I'm trying to think. Sorry, if you're saying previous trials may not be well done, could we go back and have a look at it? Is that what you're saying?
Even when they're well done, they're not always
reproducible. They should be. So as in a clinical trials unit, we validate everything. We do everything twice. And yes, we have auditable, validated processes. So there's a lot of trials that sit outside units. And you're right, that I don't know how much validation and things go on there. But yeah, that absolutely is key. Yeah.
Thank you.
The question up at the back there.
Hi. I completely agree with you in the next step about getting the trials more patient-centered. And right now it's a big wave talking about patient engagement. What do you think about the patient engagement since you are starting the drug development? Or where do you think it's better to involve the patients? Or how do you think you can make the trials more patient-centered?
So we work really hard at that now, and part of that was from the NIHR putting it at the forefront of their agenda around about 10 years ago. And when we started to do it, it was a learning curve, but I think we're quite good at now bringing in patients really early and collaborating with, because they work in partnership with us on many different aspects of a trial, and they might be from helping us choose the outcome to helping us define the research question all the way, and then during the trial really influencing maybe the consent sheets, the way we approach what is and isn't sensible. So we are doing good practices on that now, I think. We can, Our next challenge is research inclusion, which is, slightly a different one.
Yeah, so what do you mean with research inclusion? Because if you said that the next step is making the trials more patient-centered, but we have already started, which one will be like the improvement, the next step there?
So patient-centered, the other thing, we're working in partnership with patients to help us to design and deliver trials. The one that I mentioned at the end, which is about decentralized trials, that's about taking the trials to the patient, so decentralizing it, so we're not doing it in clinics and stuff, and actually we deliver the intervention to their home, and they can do their appointments at home and all those kind of things. So they're slightly different things. So that's about being patient-centered with regard to delivery, and the other one was being patient-centered with regard to working with them and making sure it's actually addressing questions they want and in an acceptable way. And then research inclusion is about, we lack diversity in who's included for the trials sometimes, and we lack diversity, one, because it might be hard to reach out to certain people, and other times because we're too cautious as well, because, for example, certainly in children, pregnant women, and babies, we don't do enough of inclusion because it's very difficult to do trials in those populations.
All right, agreed. Thank you for the clarification.
Any other questions? There's one there.
Thanks, Victoria. It's great to hear about your data-enabled trials towards the end. And I guess now we do have a lot more routinely collected data available. I guess there might be some people that say, why do you need to do a trial? Can you just do a target trial, for example? And I just wondered what your views were on target trials and if you think they'll ever replace the need for doing a randomized comparison.
Yeah. So I'm not opposed to all the work that's going into target trials, and we had a good meeting.
So do you want to just explain what a target trial is?
A target trial is where we're trying to emulate randomization. We're sort of trying to find out what would have happened in a kind of more of an observational setting. So we're trying to make it as good as a randomized control trial, but we haven't actually randomized. We've just done it. We've picked out those that we believe would have been randomized. So, and I think the methods are there, but actually I think even we had an event earlier in the year, and actually, there's a long way to go, I think, and it's really complicated. And I think ultimately, it's still an observational study in my mind. Sorry. Yeah.
There's a question up there.
So, the more adaptive and complex the trials, the greater the burden on the statisticians working behind the scenes, the more obscure the analysis and the less transparency there is around the results and the connection between the results and the data, the less replicable the whole trial is. How do you suggest we balance these things?
I would hope that, I think that we're still learning with adaptive trial designs, and I think they do take more resource. And I don't know if you're alluding to maybe trial teams that are completely separated from the data and the design team. Would you mean design team versus the analysis team? And there being mistakes and things there? Is that the thing that you're alluding to?
Right, potential for undetected errors in the analysis.
Yeah, I have had my own personal experience actually of where we did have an error on the way, which was actually we did pick it up, but there were learnings from that, Ray. But I think that is about process ultimately, and that we have to do our processes better. We shouldn't be reserved on adapting, making advances in adaptive trial design. I think we probably just need to skill ourselves up. And again, that's what we've been doing as a CTU. We've really been embracing adaptive trial designs across all of our departments in ICTU. So, sections in ICTU, yeah.
Victoria, I would never have been guilty, obviously, of thinking of the trials unit as being a bit of a bolt-on for a bit of methodology to support an NIHR application. But can I ask what you're doing to educate clinical academics who want to get into running clinical trials in their field to avoid that sort of behaviour?
Yeah, I hate to say this, Mark, but it's definitely the younger cohort coming through don't seem to be have that attitude so much. No, we do we do a lot of working with we do our contemporary clinical trials course. I don't know what we do. I think again, it's about collaboration, because when we join up straight away, we're trying, as statistical and operations, working really, really early with the investigators. So I don't know whether we're doing anything bigger than that. But I think there is an appreciation in the way that there hasn't been for many years before about that collaboration. Yeah.
Got time for 2 more quick questions. There's one there. Yeah.
Thank you very much for the lecture. I'd like to ask if we want to trial a treatment, the effectiveness of a treatment with a low number of data, because the number of data are low, what would we do as an alternative method to have a reliable set of data instead to be working it out?
It's a really, really good question, and it's like there's been many trials I've done where the sample size, like I say, I get Catherine Smith's and Sagel's, where the sample size is too small and we can't do a hypothesis test on what we believe is the minimum important difference or the true non-inferiority margin. And so, honestly, I just say, you just don't test. Because I don't understand why we bury our heads in the sand and effectively underpower trials. So what do you do instead? I showed with the Sagel's example, we took it into a non-inferiority, a Bayesian framework, and we've just saying we didn't do a power calculation even in the Bayesian framework, because we just said we can't do anything. What we'll do is we'll give you some probabilities of non-inferiority, and we'll let the clinicians whoever, the stakeholders, decide what they think about that level of probability. And now one of the things that they said to us is, well, maybe you should have done some elicitation to see what would have been a threshold for them changing their treatment decision on those probabilities. And that certainly is something we could do, but then it's just transparent. So transparency is the key. Don't hypothesis test if you're underpowered, is my message on that. And Max Palmer did a wonderful framework about small populations and all the things you need to check first, like, are you really trying, have you done a massive, are you really trying hard to recruit? And if so, test at a lower significance level if you're keeping it in the Frequentis framework. So yeah, there are lots of things. Ultimately, you can't do anything, but I would absolutely avoid doing underpowered hypothesis tests and then making errors in those conclusions, yeah.
Okay, thank you very much.
I think there was one last question at the back there.
Thank you very much. Sorry if I did not pick it up, but I was just wondering, do you have any AI help? And if yes, how do you mitigate the risk associated with it on your trials, whether it is on the design or analysis?
Yeah, so AI is something that we've started to really look at. We don't use it at all at the moment, only in some administrative tasks within the unit to help us. But we're not using AI at the moment. But In fact, we've got some conversations coming up where we will be just seeing what, just to explore what there is out there, but we're not using it at the moment at all.
Great. Thank you, everybody. And I'm sorry if we didn't get to your question. It's my pleasure now to close the session by asking Deborah Ashby, Dean of the Faculty of Medicine and long-time collaborator and colleague of Victoria's, to give the vote of thanks.
Thank you. And just seeing how many people are here, I think is testament to what Victoria does and is doing. I've known Victoria a long time. I'm Dean of the Faculty of Medicine now, but as you may have gleaned from that, by profession, I'm a statistician trialist. And I came here almost 2 decades ago, worked with Neil Poole to set up the unit. But I always knew we needed more and better statisticians. And so when we actually, BRC funded those posts originally, and when Victoria put her hat in the ring, I already knew her from the statistical world, I thought, yes, this is what we want. And we managed to recruit her, and you can see why she's gone. She came across, actually, I think, sideways as a senior lecturer, promoted to reader, and then to chair. And the science is wonderful, and you've seen some of that today. But what I really appreciate is how she does it. The number of people she has also attracted in, more than I managed to do. They have grown from research assistants to sort of senior people now. And the atmosphere in the unit, I mean, it was good anyway, but it's really, I think, gone from strength to strength under Victoria's leadership. So, however well you think you know someone, and I managed Victoria for quite a while, I had no idea about the theatre. So what I love about inaugural lectures is that you see a side of people that you don't otherwise see. So for those of you who haven't yet given an inaugural lecture, please, that's an exemplar of how they should be done. There's plenty of time for the science, and you need some of that, but actually the person is what really matters. And as I was watching that, a phrase came to mind, which is that a bad workman always blames his tools. And we get that in trials. Oh, they're expensive, they're not the right population, they take too long. Oh, what do you mean I've got to do it through a trials unit? Whereas a good workwoman just makes her own tools and then does really beautiful, elegant things with it. And now that I know that Victoria did theatre design before she did this, I'm beginning to understand why she manages to do it so well and so often in such a variety of scenarios. Whether it's sort of looking at the safety of the COVID studies or the platform trials. And don't you feel there is more to come? You can just feel her bursting with ideas and the team round her to work with. So I'm very conscious that if we're to be collaborative, we really need to get out there. We've got some drinks out there, and I'm now the only thing standing between you and those drinks. So what I'll ask you to do, my technical job is to propose the vote of thanks. Well, I'm proposing it. Please join me in thanking Victoria for a wonderful lecture. Well done.
And we'll say goodbye to those online. Unfortunately, you can't join us for drinks. But for the rest of us, it's down the stairs into the lobby of the building.